I am Judy Wiles. I'm the Director of Research and Advocacy for Defeat Duchenne Canada. We're very happy to welcome Dr. Michael Rudnicki, who is with the Regenerative Medicine Program at Ottawa Hospital Research Institute and my alma mater, University of Ottawa. He has lots of exciting research going on that he's here to talk to us about. And we're very, very proud to say that we have been supporting Dr. Rudnicki's research for a very long time. We won't go into how many years, but we'll just say it's been a while. If anyone has questions throughout, please use the Q&A function at the bottom of the screen, and we'll do our best to get to as many as we can. I'm going to hand it over to you now, Dr. Rudnicki.
Thank you, Judy, and it's a real pleasure to be here today, and thank all of you for coming out for this. So what I'm going to do is present a brief PowerPoint describing the findings from our recent publication, and plus a little bit more work from a second publication, both of which were supported very generously by the donors who support Defeat Duchenne, and we very much appreciate all of that. Shown in this slide is Ushia Garian Rodriguez. She was the postdoctoral fellow in my lab, a PhD scientist who was the lead on this project, although we have very many collaborators helping us out. She's now returned to Spain, and she's working back in that country as a scientist, so she's moved on from my lab. So let's dive right into it. So this work is about Wnt7a, which is a secreted protein that's expressed in regenerating skeletal muscle. I'll tell you more about that later. We've done a lot of work on Wnt7a. This is a protein that's secreted, so that leaves cells and then travels to other cells that have a receptor that bind it and then react. And what we discovered is that Wnt7a acts at 3 levels through muscle regeneration. It stimulates an increase in the numbers of stem cells by stimulating stem cell expansion. It stimulates the motility of stem cells and their progenitors, so they disperse and travel throughout the regenerating tissue. And it also acts to enhance the growth of the differentiated muscle fiber. And what we discovered is that if we delivered this protein by injection into a regenerating muscle of a mouse that has Duchenne muscular dystrophy, it enhanced and augmented regeneration, giving you a bigger, better muscle. But the problem with Wnt7a is that it's quite an insoluble protein. It has sugar and other modifications that basically make it very sticky. And so if you inject it, it really doesn't travel very far. And that's always been a problem about how we could use this for a therapeutic, because you don't want to be, for example, as a treatment, as a therapy, having to inject the hundreds of different muscles that are present throughout our body. It's just, that's too many needles and just not attractable as a therapy. Normally, this summarizes what I said. Wnt7a was classically thought to be secreted as a free protein, where it would diffuse to the adjacent cell or a nearby cell where you'd have a receiver cell which would respond to that signal. So it's excreted from one cell and finds its way to another cell that then responds in some way. But what we wanted to explore was whether or not Wnt7a is secreted on what's called an exosome. Exosomes are small, pinched off fragments of cells. They're small, pretty uniform in size, about 100 microns. There's a particular mechanism that manufactures these in cells and then secretes them outside. But the thing about these exosomes is that they're quite soluble, and they can carry insoluble proteins as cargo throughout the body, where it can be taken up by a recipient cell. So that recipient cell can be very far away from the cell secreting this. And also you can inject preparations of these into the tail vein or into the circulation and have it travel throughout the body. So we were interested in whether or not was Wnt7a being secreted in exosomes rather than free protein? And 2, could we harness that to deliver Wnt7a as a potential therapy? So first thing we noted is that if we damage muscle, by injecting what's called a cardiotoxin into that muscle. This makes a very severe injury. You can see that Wnt7a in green here is turned on in the newly forming myofibers, and normally it's basically off. And when we prepared exosomes, when we looked by electron microscopy, we could see exosomes, these little vesicles with Wnt7a on them. Those black spots are Wnt7a on the outside of these exosomes. So we next developed a system where we could study Wnt7a on exosomes, as well as have control exosomes lacking Wnt7a that are both prepared from regenerating muscle. So here's electron microscopy showing you the mouse which has Wnt7a, and so the exosomes from regenerating muscle have Wnt7a on them. And then if we delete Wnt7a using the genetic system, we have no Wnt7a on those exosomes. So we can compare the bioactivity of Wnt7a on exosomes or the same exosomes lacking Wnt7a. And we find in this experiment, we're just showing the induction of fiber growth by Wnt7a, but we've also documented that these exosomes stimulate stem cell expansion and stem cell motility. So Wnt7a in exosomes has full bioactivity, as they say. It works very well. In fact, it works a little bit better, or quite a bit better than just free protein. So it works. So we were interested in how is Wnt7a getting onto exosomes. So a protein can be thought of like a sentence in a book. It has a sequence of amino acids. It's built of amino acids. It's like a rod that then coils into a particular structure. So what we can do is nibble away from either end and see if we can delete the sequence that then directs it to exosomes. So in this first experiment, this slide, we deleted from the C terminus. The full-length protein is 349 amino acids, so we removed more and more. And we did the same thing from the N terminus. We removed more and more and more and more. And what we found is that the portion that's required to go in the exosomes was somewhere in the middle of the protein, which was a surprise because we know there's other things at either end that are doing jobs. So somewhere in the middle, in the center 100 amino acids was required to put this onto the surface of exosomes. So when we looked at the structure, you can see the structure shown here, it kind of looks like a clamp. And how this protein interacts with the receptor on the surface of the cells is that clamp binds that protein receptor that's on the surface of the cells, just like a clamp. And what we found is that this little blip at the back end, this loop here, you can see that, was in a region that was highly unstructured by this analysis. And this center 18 amino acids was very unstructured. And we know that signal peptides of other types, like the one that at the N terminus, are also unstructured. So that became a candidate region. What we did is we took those 18 amino acids and here we replaced them with a linker of 4 amino acids. And what happened when we did that is that it was no longer secreted as exosomes, and rather it was secreted as free protein. You can see this in the bottom. So without this 18 amino acids, boom, you lose the ability to be secreted on exosomes, and instead it comes out of the cell as free protein. So this little blip sticking out the back of the protein is somehow involved with regulating its secretion in these little vesicles.
Can I interject with a really quick question just to make sure that I'm following, if you don't mind?
Yes.
Am I correct then, just to put this into a visual for those of us who aren't quite as well versed, is this similar then, to very simplify it, this is the entire Wnt7a, and then it's almost like you're putting it into different building blocks and you're inserting something into it. Am I right?
Yes, we swapped out this 18 amino acid tag
Right.
with just a generic sequence that is just a linker that has no function.
Great. Thank you.
And when we did that, we lost the ability to come out as exosomes.
Thank you.
So this next experiment, though, how come I'm not advancing? Let's try this. There we go. Was very cool. what we did is we took this 18 amino acid tag, a very, very short piece of a protein, and we linked it to a different protein that's normally not on exosomes. And this protein is called halo. This is a bacterial protein that's used pretty widely in cell biology to tag proteins so that because what it does, it binds a dye that you can then visualize in the microscope. So you can follow where proteins go. Normally this protein by itself, it's a bacterial protein, just sits in a cell and doesn't go anywhere. It's not secreted um, uh, and it just hangs out. But what we did is we stuck on our 18 amino acid fragment onto halo. So it's halo EBP. And we put it into cells and we found it was secreted on exosomes. We could isolate media from those cells, so that everything that's been excreted, put that media onto a receiver cells, and you can see in the bottom right, those receiver cells would take up the tag that was on the exosome. So this proves that this tag is what's directing proteins to exosomes and allows their take up by exosomes and then transfer to a different cell. And this has big implications for putting any protein you wish onto the surface of an exosome, including proteins that could direct binding to specific cell types or specific tissues, which is a really interesting idea. We were next interested in what was this 18 amino acid address interacting with. So we did an experiment to understand which proteins it was interacting with. We identified candidates. We showed those candidates were on exosomes, and a collaborator in Spain, Aitor Hierro, actually did crystal structures and mutagenesis assays to identify exactly how the tag was interacting with this protein. And this protein is called, it's a codomer protein, it's called COP-B2. And so this just shows you the structural stuff, which is really kind of pretty. So this green donut shape is this protein called B protein. It has a hole in the center. And this orange sequence is a sequence of the tag showing exactly how it's interacting with the donut to achieve binding. And you can see in this structure in purple is full-length Wnt7a. There's our clamp. And the EBP sticks out the back and it sticks into the hole of the donut of COP-B2 so that it's presented on the surface of exosomes so that the clamp is available for binding the receptor. Isn't that cool? If I could put it that way.
It is very cool.
And
Thank you.
this is a series of experiments where we mutate the different amino acids that are believed to be interacting and show that if the critical ones are the ones shown in red, it's this kick motif. These are 3 lysines. And if we mutate either of these 2 front ones, we prevent binding to the protein. So that's an important biochemistry to understand the mechanism by which this Wnt7a is binding the protein. Now, does this have relevance to the function of Wnt7a in the animal? So we wanted to ask whether if you can't be secreted in exosomes, can you still stimulate regeneration? Or do you require the ability to be secreted on exosomes for this activity? And indeed, you do. So if we overexpress Wnt7a in a regenerating muscle, we have augmented repair. If we overexpress the mutant, lacking the ability to secrete on exosomes, it doesn't work. In fact, you have impaired repair, which is quite diabolical because that means that you have to be secreted on exosomes for proper biological activity. And I should add, as an aside, that the recombinant protein by itself, when we test it in cultured cells, it has full activity. It works very well by itself. But in vivo, in order for it to travel the distances required for activity, it has to be on an exosome. Now, what happens if we give it to a mouse lacking dystrophin? So this is the so-called MDX mouse. Now, for this experiment, we built a sensitized mouse lacking Wnt7a. So these mice have have already impaired regeneration because they lack Wnt7a. But our rationale here was that it allowed us then to test whether or not we could deliver it systemically to see an effect. And sure enough, if you look at fourth generation, exosomes carrying Wnt7a, a single injection into the venous system resulted in a muscle that could generate more force. So this implies that we're enhancing and augmenting regeneration in this disease model. Now, right now, we're just testing it in multiple injections of exosomes in an MDX mouse to see whether this can ameliorate disease progression, of course. So those experiments are in progress. So that was with a muscle where we did an acute injury with cardiotoxin, but we were also interested in other muscles which hadn't been injured with chemical, just in the regular MDX mouse, again lacking Wnt7a. So we looked in the diaphragm. So across the top are mice injected with control EVs lacking Wnt7a, and we see the normal dystrophic muscle. These are 3 different diaphragms showing you different areas of fibrosis. And if we looked at the Wnt7a EV treatment, we saw this rather dramatic reduction in fibrotic areas, suggesting that we've really augmented repair in these mice. So together that argues that yes, we can deliver Wnt7a systemically in exosomes, and it can have a positive therapeutic effect. In conclusion, Wnt7a secreted in exosomes. We've identified an 18 amino acid tag that acts as an address for the trafficking system to load this on the surface of exosomes for secretion that can then go long distances throughout the body. This tag can be linked to any protein to put it on an exosome, we believe. And exosomal Wnt7a can be delivered through the circulation, through the blood system, to get to all muscles in the body and potentially have a positive therapeutic effect. And here are the people who did the work. Of course, Uxiel was a postdoctoral fellow, first author on the papers. She was supported by Hong Ming, Isan, Fan Zhao, David Datsku in the lab to get the project done. And we also had a whole series of collaborators that are listed at the bottom. In particular, Eito Iero did the crystallography. Thank you very much.
Thank you so much for thank you so much for that lovely presentation. Can we loop it in to specifically to Duchenne? So do you foresee that as being a promising treatment? How do you see that moving forward? And if anyone else has questions, please feel free to drop them into the Q&A and we'll do our best to get to them.
So there are multiple approaches being explored clinically as a treatment for DMD. We are also, as you know, I'm a founding scientist in Satellos, where we've developed a drug, a pill, that mobilizes muscle stem cells and we believe stimulates regeneration. That looks like a very tractable, easy way forward. This approach is really interesting and has potential. I think, however, it will be expensive and difficult because it will require repeated treatments on an ongoing basis throughout life, and to manufacture these exosomes, I think will be expensive. But we also have to evaluate how effective it is over the long run. And in doing it in a large animal like a human being would require a lot of material.
For sure. I think you've already answered the one question that has come up is with your answer to the Duchenne question, if it would be applicable for other muscular dystrophies. Quite likely the answer would be yes, but similar limitations. Somebody else is asking, but I suspect they are referring to your Satellos clinical trials because they're asking what is the status of starting the clinical trial in Canada. So I don't know if you want to take a moment and speak to Satellos and your plans there.
Yes. So we've been making really quite amazing progress in Satellos, and maybe we should do another webinar on that topic. But
we definitely should.
We definitely should. So the phase 1 trial, which is the safety trial, is well underway. They've dosed the first DMD patients. That trial is all about safety. So they're doing a lot of toxicity readouts and so on. And so far, the drug is very well tolerated, totally safe, no issues whatsoever, which looks really good.
Great.
But in the phase 2, which is testing how well it works for treating DMD patients, will be starting in the new year. And there'll be several trials there occurring in Canada, the US, Australia, and the UK, and possibly in Europe. Those phase 2 sites are being settled on now, and there'll be, I believe, at least 2 sites in Canada that'll be involved.
Great.
But that's still being developed. But certainly for me, I would really like to see lots of sites in Canada involved.
So would we.
But uh, so that's all in progress and being developed. But there will be Canadian sites, but I don't know exactly where those will end up being at this point.
Great. We have another one. Let me just quickly. So the question is, in the phase 1 clinical trials that have been completed thus far, is there any information regarding the effect on inflammation, signals or biomarkers, or any effects on physical performance in the healthy patients who participated, even if anecdotal?
The readouts were around safety, so heart function, kidney function, et cetera, et cetera, et cetera, liver. It was only for 3 weeks, I believe. So it's really too short to evaluate effectiveness. And it wasn't designed to do that. The way
Right.
that regulators work, phase 1, they want to make sure it's safe first before they proceed to a vulnerable population, in this case, DMD patients.
I would also suspect that you wouldn't necessarily, the results in healthy volunteers would not be what you would expect to see in DMD patients anyway. So
So,
any signals that you might see would not necessarily be applicable. Correct me if I'm wrong.
the final arm is adult DMD patients. So I believe there's 10
Oh,
adult
great.
DMDs who are also getting the drug, but that's again to ensure that it's safe and that the pharmacokinetic data is the same as a healthy person, which we anticipate fully that it will be. And so far, it looks fine.
That's great. Does anyone else have any questions? I think that's it for now. Well, thank you so much for taking the time to go through this with us. I definitely agree that we should do another, and we can focus on Satellos if you like, and we'll definitely announce that beforehand so everyone can get on board. Thank you again. Please, we'll be in touch shortly. Thanks, everyone.
Thank you, everyone.
Have a great day.